compounds that are produced by plants that they use to facilitate the interaction with their environment. These compounds are often things that humans are familiar with, regardless of whether you're aware of them or not. This includes things like compounds like piperine or capsaicin that we use as spices, or compounds that give plants color, like anthocyanins or betalains, or even compounds that humans use as medicines, like morphine or Taxol. They're all around us and play an integral role in our lives. So many of these specialized metabolites are produced in specialized cell types. The idea is to partition these compounds away from the rest of the plant. Oftentimes, these compounds, when accumulated in high enough amounts, can damage the plant cells themselves. So the idea is to sort of keep them away from the rest of the plant, and then to localize them to areas in which they'll interface with other organisms. One prominent form of these specialized cell types are glandular trichomes. These are epidermal structures present on the surface of many different plant species, and they often serve the tips of those glands. You can see are metabolic hubs for many different types of specialized metabolites. They serve as a site of synthesis for specialized metabolites like terpenes or acylsugars in the case of our lab. Acylsugars, as I mentioned, are a group of specialized metabolites that are produced in glandular trichomes in plants across the Solanaceae family. These molecules typically possess a sucrose core that's then conjugated to a variety of different acyl chains, as you can see here. Now, the molecule on the right is one of the prominent acyl sucroses produced in cultivated tomato. Now, it contains a sucrose core, as you can see in the nomenclature, a sucrose core with 4 acyl chains conjugated to it and 17 carbons in those acyl chains. So we then depict each of those individual chains after the S417 there to depict exactly what the makeup is. Now these acyl sucroses, these acyl sugars are synthesized through the action of a group of enzymes called BHD acyltransferases, or we refer to as acyl sugar acyltransferases. They take sucrose or some of these acylated intermediates and generate a molecule similar to what you see on the right there. Now these ASATs are expressed in the tip cells of those glandular trichomes. And we know this because we've studied a number of their promoters and found they all express in the tip cells of those glandular trichomes. Now, for us, it was particularly interesting when we moved beyond tomato. And the reason that was the case is because there's a tremendous amount of acyl sugar diversity present across the Solanaceae family. Now, all of the acyl sugar components I'm going to show you are derived from these 7 species on the slide. Now, the acyl sugar diversity starts with a sugar core, whether it's relatively common sugars like sucrose or glucose, or a little bit more unusual ones like myo-inositol or inositol sugar conjugates. This diversity in the sugar core is further complemented by the variation in acyl chains that we see that are often derived from acyl chains from amino acid metabolism or fatty acid metabolism, or even from other parts of metabolism, for example, in the case of like phenylacetate. But this omits differences in variation in the acylation patterns that we see across species that is a product of the differences in enzymes. Now the question becomes, well, why are we interested in acyl sugars, which I haven't talked about? As it turns out, they play a role in plant defense against insects. Now, it was previously thought that acyl sugars are simply sticky stuff that the plants produced on their surface. But more and more studies have come out and demonstrated that there are differences in variation in arthropod mortality that's dependent on the characteristics of the acyl sugars. Paterka et al., for example, found variation in the mortality of these 3 different insects that depends on the characteristic of the sugar core and acyl chains. Zhao et al. Found similar findings in terms of the variation, but they looked at gram-positive bacteria, the effect of acyl sugars on gram-positive bacteria. Finally, Leong et al. Studied these 2 insects and found a combination of acyl sucroses and glucoses were more effective than either of the independent acyl sugars. And so that's one of the reasons why we're interested in trying to study acyl sugar diversity across the Solanaceae family. My thesis work primarily focused on characterizing acylhexose biosynthesis in the Solanum genus. I'm not going to talk about most of the other work, but I'm going to focus on one particular species. So it spanned multiple species, but I'm going to focus on Solanum ketonense in today's talk. So I'm going to describe the characterization of a handful of different enzymes in Solanum ketonense and their role in acyl sugar biosynthesis. Okay. As I mentioned, the species that I'm going to talk about is Solanum ketonense. This is a fruit crop from South America, typically referred to as lulo. So this fruit crop produces these small orange fruits that they often use in drinks. But while that's interesting, we are particularly interested in the acyl sugars that this species produces. The acyl sugars contain a myoinositol core. Now, this is a type of acyl sugar that at the time, we hadn't seen either in the tomato clade or more broadly across the Solanaceae we had characterized. Now, this myoinositol core is further decorated with acyl chains that contain 2, 10, and 12 carbons in length and seem to be derived. Other work indicates that it seems to be derived from fatty acid metabolism. The acyl sugar complexity is increased by what we further glycosylation of that central myoinositol core, where we see the presence or the glycosylation with glucose, N-acetylglucosamine, and xylose. We are particularly interested in trying to understand how the plants are synthesizing these compounds. Now, at the time, the resources for Solanum ketonense were essentially non-existent. There were no genome sequence available, no transformation technology. And so Gaurav Mogi, a postdoc at the time, was actually interested in studying acyl sugar diversity across the Solanaceae family. And he picked Solanum ketonense as one of those interesting species due to the acyl sugars that it produced. Now, Gaurav took advantage of a trait that's very common for specialized metabolites, and that they're produced in specific tissues. So you can use the expression of the acyl sugar producing tissues versus non-producing tissues to try to find these pathways using RNA-seq. So he performed RNA sequencing on glandular trichomes and the underlying petiole tissue in order to try to identify putative ASAT candidates. I dug into his RNA sequencing data, and we were able to identify a number of proteins, or basically tried to identify proteins that match previous BHDs, including similar protein size, motifs, both in terms of having the correct ones and the orientation of those motifs. We then restricted candidates to those that were expressed in the trichomes, which would make sense for acyl sugar biosynthesis, and then performed phylogenetic analysis and focused on candidates that were closely related to existing acyl sugar enzymes in other species. So one of the enzymes I'm going to talk about was related to ASAT4 from tomato. Now, ASAT4 is an enzyme responsible for catalyzing the activity that you can see in the bottom right, where it's adding an acetyl group to the R2 position of the 6-membered ring in sucrose. Now, this enzyme, this ASAT4 homolog, was one of the most abundant transcripts that we observed in the RNA sequencing data. And so that, in combination with the relationship to ASAT4, is what got us excited about this enzyme, as well as the high expression and the enrichment in trichomes. Okay. As I mentioned, there was no transformation technology available in order to test our candidates to knock out these genes. So what we did is we employed VIGS, or virus-induced gene silencing. This is a technology that transiently silences some of these target genes. So essentially, you take part of the cDNA and you express it in a viral vector, and you essentially trick the plants into reducing expression of their endogenous genes, and then you screen for phenotypes. We used phytoene desaturase, which results in a photobleaching phenotype when silenced, in order to guide the sampling of our plants in Solanum ketonense. We then examined the acyl sugars in some of those plants. Now, the first thing that I want to mention is that the major acyl sugars in Solanum ketonense are primarily tri- and tetraacylated inositols. They differ primarily by the presence of this acetyl group. You can see here. Now, what we found is when we targeted the ASAT4 homolog, we saw a substantial change in the ratios of tri to tetraacylated inositols. So essentially, presence or absence of those acetyl groups. We saw this trend was maintained across a number of different plants in which we saw increases in I322 to I424 and 324 to 426, differing by a single acetyl group. Based on this, we hypothesized that in fact, this enzyme was a triacyl inositol acetyltransferase. This is what I'll refer to it as going forward. Okay, so we had shown in vivo that seemed to suggest that this enzyme was involved in acetylating triacyl inositols. If that was true, we hypothesized that the enzyme would be able to use the triacyl inositols in vitro. We purified this I322, this triacyl inositol from the plant, and we heterologously expressed TAIAT and found when we combined them in a tube, we saw substantial conversion from the triacyl to tetraacylated inositol, indicating that TIAT is actually catalyzing the acetylation of this in vitro. We then took that and compared it to the in-plant acyl sugar just to verify the identity. And what we found is that in fragmentation, that I'm not showing here, but in coelution, which I am, it coelutes nicely with the in-plant acyl sugar, indicating that it's consistent with a role in plants. We also found that in addition to that acetylating I322, the enzyme was able to acetylate I324, the other major triacyl inositol that I mentioned. So for all of the triacyl inositols, this enzyme seems to be at least able to do that activity, but we think that it is the primary enzyme doing that. Okay, so I talked a little bit about TIAT, which is catalyzing the acetylation of those triacyl inositols. We used VIGS and in vitro biochemistry to demonstrate that, but much of the remaining pathway was uncharacterized. And so we're trying to identify other enzymes that could be involved in this process. So we went back to that RNA sequencing data and we focused on other potential ASAT homologs. We worked on the assumption that, or our thought process at this point was that acyl inositol, the fact that TIAT was catalyzing similar activity to ASAT4, its phylogenetic relationship, and the presence of the pathways and trichomes indicated that these two pathways were evolutionarily related. Now, there were a handful of other, if we operate under that assumption, there are a handful of other ASAT homologs that could be involved in acyl inositol biosynthesis. Now, all three of these homologs were both expressed and greatly enriched in the trichomes relative to the non-acyl sugar producing petiole tissues. These acyl-CoA, or these enzymes, use different acyl acceptors, but we're interested in trying to see if in fact they're catalyzing these acyl inositol acylating activities. So we went back to our trusty VIGS experiment. We targeted this homolog of ASAT1, and what we found was a common phenotype that we observed before, basically a reduction in total acyl inositol, so we quantified relative to the empty vector plants. And this ended up being a common theme when we targeted ASAT3 and another ASAT4 homolog. We saw reductions in acyl sugars across the board, indicating that at least one or more of these enzymes may be involved in acyl inositol biosynthesis. Now the question then becomes, what steps do each one or more of these ASAT homologs catalyze? Now the VIGS suggested that the ASAT homologs are in fact involved. We expected that if we put these enzymes together, we'd see one or more of these acyl inositol intermediates. And so when we combined ASAT 1, 3, and 4 together, what we found was not in fact an intermediate so much as we found a triacyl inositol, at least one of the products that accumulates in vitro. So the triacyl inositols do. Now, unfortunately for us, what we found is that the in vitro products did not coelute with what we with keto N-C-acyl sugars that we had extracted from the plant itself. So it's quite similar to the implant acyl sugars, but we think that the acylation pattern is slightly different. Now, this is still interesting to us because essentially these enzymes are still catalyzing these activities in vitro. So we were interested in trying to determine exactly what enzymes are responsible for catalyzing what steps. And it's not representing the final or the complete implanted pathway, but it's still a way to synthesize these acyl sugars and potentially test them for other purposes. So we went about trying to characterize some of these enzymes. The first was this homolog of ASAT1. It is a natural fit for the first step in this pathway. The reason is that in tomato, this SLASAT1, its closest homolog, or one of its closest homolog, catalyzes the acylation of sucrose with a C5 acyl chain. Now, there are no C5 acyl chains, but what we found is when we supplied this ASAT1 homolog with myo-inositol and a straight 10-carbon acyl chain, acyl-CoA, that the enzyme is able to catalyze that acylation in vitro. We've done much more characterization of this product, including using it with another enzyme. So this ASAT4, we found that in addition to catalyzing that first reaction, when we add the homolog of ASAT4 together with that first enzyme, we begin to accumulate this diacyl-inositol I212. Now, this is one of the two potential intermediates to catalyzing a triacyl-inositol. But what was interesting is that this enzyme is able to use acetyl-CoA to catalyze that second acylation. Now, this is consistent with other ASAT4 homologs in which they typically use acetyl-CoA, but they're generally localized towards the end of the pathway. So the acyl acceptor is different. Oftentimes in tomato, for example, it's acylating a triacyl sucrose that I mentioned. But this enzyme seems to have shifted forward in the pathway, or at least in the in vitro path, its ability to use these substrates shifted forward in the pathway. Finally, we're able to, based on those in vitro results, we hypothesized that this ASAT3 homolog would catalyze that third step in vitro. Consistent with that, we found that when we ran sequential assays, that when we added the ASAT3 homolog last, we began to accumulate the triacyl-inositol that was absent when we swapped ASAT3 and the 4 homolog. So if that makes sense, essentially, if we add two enzymes, ASAT1 and 3 together first, and then 4, we don't see the final product, that I322. Whereas when we add 1 and 4, kill the reaction, and then add 3, that is when we begin to accumulate this final in vitro product, indicating that in fact, that's the order of acylation, consistent with our previous assays. So just to summarize a bit, there seems to be, or we have a conflict between our in vitro and implanted data that suggests a missing factor. The VIGS data clearly indicates that the ASAT homologs are involved in acyl-inositol biosynthesis, whereas the in vitro data suggests some placement of these enzymes in the pathway, at least the ability to use the substrates or intermediates we would expect, but some piece is missing in trying to reconcile what we see in vitro and the implant acyl sugars. What is clear is the evolutionary relationship between acyl sucrose and acyl-inositol biosynthesis. We see that both of these pathways are clearly happening in glandular trichomes, as well as the in vitro and in vivo data suggesting that one or more of these homologs are involved. Okay. Wanted to thank my PI, Rob Last, for helping me work through this pathway and my PhD research, as well as my lab mates and committee, and then the funding agencies for NSF and NIH for funding me. So I can take any questions.
Thanks, Bryan. If folks have questions, please feel free to raise your hand or pop a message in the chat or whatever you'd prefer. Looks like Jorg's got a question. Go for it, Jorg.
Thanks. That was a great talk, Bryan. Really enjoyed it. I have a totally naive question. What does acyl sucrose taste like to the human perception?
Oh, I think the answer is soap.
This tastes like soap.
Kind of, yeah. It's yeah. I'm not sure if Gaurav would be able to answer this question. He might have done that experiment.
Okay, but it does not taste like sucrose, not like sugar.
No.
And in the human digestive system, would we be able to break it down to release sucrose from it? Yeah, I think Bryan has lost his mic.
I think he's working on it.
Yeah.
Hello?
Yeah, you're back, Bryan.
Sorry about that. My audio cut out. Thanks. You were talking about digestion in the human tract.
Yeah.
I'm not 100% sure about humans, but I know that in insects, it's been shown that essentially you're hydrolyzing the acyl chains from the actual sugar, essentially, and so they think that may be part of the mechanism of some of the toxicity to the insects or the protection against insects is that they digest it, and then the free fatty acids are what actually can affect the insect.
Great. Thanks, Bryan. Great talk.
Thank you. Dr. Barkman.
I don't know. Marchman.
Hi, Bryan.
In the chat, or shall we like? Oh, is he talking?
If I could just ask verbally, it'll save me the pain of trying to type out my question. So that was interesting talk. I'm wondering if your emerging picture is that what's conserved in your ASATs is the acyl donor, but then what's changing in your enzymes is the sugar that they're ornamenting. Is that the picture I'm getting?
Yeah, so that's definitely part of it, right? So you've essentially shifted from using, so in tomato, they primarily use sucrose, right? In a number of different species, they're using sucrose as the acyl acceptor. The shift from, so there's sort of two aspects, right? You've shifted to a slightly different sugar in myoinositol. So there's that the confirmation of that ring versus sucrose, but then the other thing is that you essentially, what we see in the case of ASAT4 is a shift in terms of the number of acyl chains that are present on the substrate that it uses, which we've done some preliminary work with and seen there's not usually a ton of crossover in terms of like using a mono versus a diacyl sucrose in this case. They seem to be generally pretty specific.
So then are you able at this point to look at a clade of sequences and say, I don't know what sugar they're going to bind, but they're definitely going to add decanoic acid to the sugar or something like that? Are those kind of rules emerging at this point?
To some degree. So I didn't show it, but for example, the previous work from our lab, the tomato ASAT1 actually does use that same C10 acyl-CoA.
Oh, okay.
We're not 100% sure. So there's a bunch of factors that sort of complicate things because dead end products can to some degree be broken down by the plants. There are enzymes that catalyze those activities. But yeah, some of our previous work demonstrated that they will in fact use that ASAT1 will use that C10 acyl-CoA. So it does seem like there's some conservation, but I think we probably need more broad characterization to really pull out those rules. But
well,
thanks.
Well, that's hopeful. It seems daunting, but it's great to see that you're some solidifying concepts here. So thank you.
Yep, thank you.
Michael, did you have a question?
Yeah, we have some questions here. You want to? You can ask the same question. Yeah. So, can you hear me well? Is it
Yep.
fine? Okay. Yeah. Thank you for the talk. So I guess this is kind of building off that previous discussion we just had. There I just wondered if you could comment a little bit on the acceptor substrate, not the donor, but the donor acyl, but the acceptor substrate specificity. So you're looking at, you showed that maybe this other one that can acylate sucrose, I believe that you showed at some point, can also do another one. What about the triacylinositol enzyme that you have? What if it encountered inositol on its own rather than a triacylinositol or something like that? Is it able to acylate that? And then I guess the sequence after that is, I guess, can you comment on what is missing in this pathway for the kind of specificity that you see within the plant? Because you mentioned that, you mentioned that the whole pathways in this glandular trichome. So what is the thing that's allowing the specificity of specific acyl sugars to emerge when we know maybe this enzyme can hit multiple spots on inositol or other acyl sugars? Is it temporal regulation? So it's a big one, maybe, but yeah.
Okay, so let me. We've done some testing with that homolog, the TIAT, the one that's able to use the triacylated inositols. It doesn't seem to use sucrose, or inositol, the unacylated molecule. We haven't tested it thoroughly with some of the intermediates, just because of it becomes very difficult to get enough of those to really do proper enzyme assays. So it's sort of like we're putting enzymes together to be able to, we see small amounts of peaks, but essentially the quantifying or getting enough to properly test of like the di and triacylated inositols outside of the in vitro, the implant acyl sugars is difficult. But it's something worth testing, because in principle, you might think that it may be easier to sort of, for the enzymes to move forward in a pathway, right? So for example, as the substrate becomes less sterically hindering, it may be easier for the enzyme to accept some of those to move from like a diacylated substrate to a monoacylated substrate, because essentially just less, the molecule is smaller, right? And so you may be able to still bind those, like the less acylated molecule. But yeah, it's a good question. I mean, we're still, to be honest with you, I think Rob's lab is interested in trying to crystallize some of these proteins to get at some of these questions, because we are interested in trying to shift substrate specificity. In terms of trying to produce, to sort of reconcile what we see in vitro and why we're producing specific types of acyl sugars, we're not 100% sure. There are enzymes responsible for, so there are acyl sugar hydrolases that are responsible for removing acyl chains from certain positions of different acyl sugars. And so there's a study published in 2015, I think, from our lab, that essentially found that when we introduced an acyl sugar hydrolase from this wild relative of tomato to tomato, they demolish the acyl sugars that accumulate in the plant. So it seems like they may be a factor that's responsible for sort of maybe editing or degrading some of the acyl sugars. So some of these things may be dead end products. Essentially, they can't be used by other enzymes, and so you have some of that being recycled. But it's still a bit of a murky picture, to be honest. We were a little bit surprised when we put these enzymes together, and we found that we got a product, right, that looked quite similar, but it's slightly different. So we're not 100% sure what's going on there. But
yeah, thank you very much. I mean, the other thing we were just discussing here was, I mean, it's always fun to talk about, but who knows how easy it is to show. I guess if there is any promiscuity to the enzymes, maybe some sort of metabolon might be involved or something like that as well, not temporal regulation or that extra player that you now just mentioned. That's really cool. Well,
yeah. I mean, it's interesting. I never sort of lean towards that, because it's sort of, I mean, it seems like it's the exception, not the rule. So, but it's, yeah.
Also, so when you said that some of these are co-expressed or whatever, they're both co-localized to a certain tissue, how tight is that co-expression or co-regulation? Are they really, really tightly going up in expression level together in the exact same spot, or,
I mean, we haven't done time courses. What we found is they're typically pretty well expressed in trichome tips, and another thing that we're working on is trying to identify transcription factors that are regulating these things. Part of the issue, though, is that potentially what we may have going on is that some of the transcription factors that are regulating the development of the trichomes are also regulating some of these biosynthetic pathways. And so trying to decouple those two things is potentially difficult because you just abolish the trichomes if you knock it out instead of the biosynthetic pathway. But again, Rob's lab is digging into that, I think. So, but yeah, they seem to be all pretty nicely co-expressed, and that's one of the other things that we're using to try to identify enzymes. So, any other questions? Dr. Barkman.
Yeah, Brian, please just call me Todd, and if you're comfortable. But
Okay.
I'm so glad to hear this story because we were trying to piece together a caffeine biosynthetic pathway, and we generated a transcriptome, and we've got a missing player. And so I just assumed our bioinformatics were insufficient, or we should have done deeper sequencing, or we should have, whatever. I don't know. And so, what do you think is the case with your missing player? I mean, obviously a genome, maybe a genome would explain or provide the missing player of your pathway, but, and I know this was sort of the question that was asked a second ago, but do you think that are transcriptome assemblies always accurate in this regard, or what?
Yeah. I think the problem for us is that we're not 100% sure what to look for. Like we've pieced the pathway together, and so we've looked, for example, for a number of BHD acyltransferases and tried all kinds of different combinations of those enzymes. The problem is I'm not sure we know enough about broader acylsugar biosynthesis and like sort of what factors are at play, right? So I mentioned these acylsugar hydrolases before. They seem to be involved in degradation / editing, but we're not 100% sure what role they actually play. And so, I mean, that's one of the things that we considered, but then it's a question of like, there's a number of different potential enzymes that could serve that role in the RNA sequencing data. And so we found that when we put these enzymes together, actually they tend to demolish the acyl sugars at the concentrations, like I think a microgram is what we were adding. But like, so the problem becomes, we're not 100% sure if it's something like that where we need to find, like there's something in the cellular milieu that we're not accounting for, in which case it's like, it's not easy to tease apart.
Well,
yeah.
I mean, we wasted months on trying to express an alternatively spliced transcript and see if that protein possibly filled in the missing role, and we're left with nothing. So anyway, I'll look forward to any insights you have. Thanks.
Thanks for the discussion.
That's an interesting question. There's a question here in the chat from Paul, which I'm happy to read. Otherwise, Paul, if you want to ask it directly, that would be completely all right as well. I'm happy to read the question.
Yeah, I could say it real quick.
Yeah, go ahead. Thank you.
See, acyl sugars do not have branched acyl chains. I saw in the tomato ones you showed the branched acyl chains. Is this because of enzyme specificity, or is there some other reason you're not observing branched acyl chains in keto NC?
Yeah, it's a good question. So I've done some rudimentary testing with some of these acyl-CoAs, so some of the shorter, like amino acid related, so like iso branch C5 or anti-iso branch C5, and found small amounts, very, very trace amounts of activity with those acyl-CoAs compared to, for example, the straight chains, 10, 12 carbons seems to work with at least some of them. So enzyme specificity definitely plays at least part of a role. My guess is that they're also probably not producing, but even when the enzymes aren't amazing at the activity, we can often see some acyl sugars that have some of those in other species. And so the other part of my thinking is that I'm not 100% sure, but we're probably not producing some of those acyl-CoAs at concentrations sufficient to synthesize those products. So it's a mixture of things.
Other questions for Bryan here?
I have another one. If you would, if there's no others right now, I can ask another one. So we're at
Please go ahead.
we're at the Max Planck Institute for Chemical Ecology here, so I got to ask some chemical ecology about the anti-herbivore defense aspect of these compounds, and you're the expert, and I don't know anything about this, so I'm just asking this purely out of I'm just curious. What is the localization of these compounds? Is it very strict to these glandular trichomes, or where else are these acyl sugars found within the plant? Because I noticed that obviously you had, it interests me very much, is that it had some effect on phloem feeders, and this has been noted for some time. So besides surface contact, is there any other way that these phloem feeders are coming in contact with these compounds?
As far as we know, it's primarily not produced in the underlying tissue in the leaves themselves or stems. Now, there's some interesting work, I think, out of Asaf Aroni's lab that they may be produced in some root tissues. Now, again, it's but they're, yeah, it seems to be primarily localized to trichomes, as far as we can tell. There's some indications of root trichomes, essentially, in some of their data. So essentially, they're producing them in the roots, but more or less in trichomes localized to those tissues. Now, yeah.
So yeah, so we don't expect them to be, I mean, yeah, how much does this, I think I should know this, but how much does this acylation affect the solubility of these sugars?
Yeah. I think substantially, right? Because I mean, in principle, you're taught, so sucrose, like, I mean, sugars like that will be highly soluble in aqueous solutions, right? But you're adding substantial hydrophobic characteristics to those molecules. So part of that may be to sort of resist those compounds washing away, right? But yeah, you're substantially reducing the solubility in aqueous solutions. So we often, in order to extract them, we have to use organic solvents. Water's not enough. So.
Okay. Yeah, no, I mean, it's interesting because the fact that it shows like this nice deterrence to phloem feeders and the like is it's interesting to me, and I was curious about if there's anything known about the transport or how it's actually getting to them or how it's affecting them. But I guess from your perspective, it would be a surface contact kind of defense.
Most likely, yeah.
That's what's doing it. Thank you.
Yep.
We've got a few more minutes for questions here. If anybody has questions about Bryan's talk or other topics as well. I guess regarding other topics, I do have a question. So Bryan, you spoke about work that, at least my understanding is this work that you performed in Michigan, if that's correct. But now you're in Florida.
Yeah.
Do you want to just give us a super brief overview of what you're working on there, out of curiosity?
Yeah. So basically, so I spent my PhD studying enzymes and specialized metabolism. So I'm interested in trying to add a flavor of synthetic biology to that. So one of the technologies that's been fairly recent in development is continuous directed evolution, with the idea being basically trying to engineer enzymes, but using in vivo selection to try to select for better variants and better, basically to be able to traverse fitness landscapes to improve enzymes. And so basically my NSF proposal was combining using continuous directed evolution with CRISPR-Cas9 mediated base editing to essentially engineer better enzymes. So I'm working on herbicide resistance and amino acid related metabolism, and then trying to basically engineer those plant enzymes and reintroduce them into plants using base editing technology. Yeah.
Interesting. Thank you. Other last questions for Bryan? Otherwise, maybe we'll call it a day. Although if folks want to stay on for further sort of more casual conversation, that would be okay as well. Alrighty, well, everyone, please remember we've got another talk in a couple of weeks from Christine Diepenbrock. We'll be sending emails about it. Please don't hesitate to tell your friends, colleagues, students, supervisors, or associate deans about phytochem talks, and we'll see you back here in a couple of weeks. Let's give Bryan one more round of applause here for a great talk. Thanks, Bryan.
Thanks for coming.
See you later, everyone.